to the weekly Bioanalysis, a KCAS Bio podcast.
Hello and welcome to the 96th episode of the weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for around 30 years. And I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I've been in the ligand binding assay and biomarker space for a little over 30 years as well.
Dominic and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a special guest, Brad Nawa, West Region Director at Alamar Bio, as part of an action-packed main topic discussion. We are thrilled to have you listening to the 96th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the weekly bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, then a focus on the topic of our choosing, before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 96, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, so as we always do in our podcast here, we'll go over some of the latest news and resources in the medical field. And then we'll jump into our main portion of things. And as John mentioned, we've got Brad from Alamar here. He's going to talk about the NULISA, which is a new ultra-sensitive approach for protein quantification. Brad will go over a little bit of his current role and previous history. Then we'll jump right into the introduction to the NULISA platform, some of the applications for the NULISA platform, really some of the advantages of it, and what the future lies for the NULISA system. We're really excited to have Brad here, and with that, we can just get going right into the news and resources. John, what's our first topic?
So, basically this has been a quiet month or so, at least regards new news. So my philosophy of never throwing anything away is somewhat paid off. This is actually a few months old, but it's still interesting in terms of the work that was being done and what could potentially help people in the long term. So this was vaccine protects mice from allergic reactions for a year. An inverse vaccine that delivers deactivated allergens to the liver was able to reduce reactions in mice with allergic asthma for at least a year after treatment. There's hope that if this translates to humans, then maybe you get a vaccine to reduce allergic reactions. So they actually obviously sensitized mice to either an egg protein or dust mite protein, and they saw that airway inflammation and mucus levels reduced after exposure after being given the vaccine. And this was, like say, early in the year and reported in Science Translational Medicine. And actually for mice with the established egg protein allergy, the protection lasted for a full year after 2 intravenous doses of the vaccine separated by a week. It's one of those things there. Sorry, I will try and say that again.
It's all good.
I'd say this is proof of concept in mice. Hopefully, they can look at a way of translating it to human. And it's the concern with something. I guess you're exposing someone to an allergen. If you're not careful, you can trigger an inflammatory response. You got the danger of anaphylaxis, etc. So what is done here is they actually added mannose sugars onto the allergens so that the protein structures are altered in the places that cause an allergic response. So then this turns the allergen into what the scientists call a toleragen. And aiming for the liver, the liver then produces long-lasting T cells, which can help calm the overactive helper T cells that spark allergic reactions. And so that's why they sent these amended allergens there. And once in the liver, the toleragens induce the organ to make regulatory T cells that quell the response to that specific allergen. So it's quite an interesting mechanism the way they're doing it. And I'll pass it to you if you want to.
Yeah, no, it is fascinating, John. Right? Like any time the word, I think that what they're trying to say is they're breaking tolerance. Which, by the way, the mechanism behind how we tolerize things,
Mm-hmm.
in
Yeah, they are.
immunological perspective, is incredibly, I don't think well understood. But I could debate that with people. But if you could tell me the exact mechanism of how a woman tolerates a baby, then you'll win a Nobel Prize. All right.
Yep, 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 yep.
Yeah. So one thing, maybe I don't know, John. Sometimes I get in good moods, bad moods. I don't know, but I don't really like the term inverse vaccine. It confused the heck out of
Well,
me. What do they mean?
it says why they're calling it an
I know,
inverse
but
vaccine.
it didn't sit well with me, right? So
Yeah,
they're trying to say they're instead of like inhibiting a bad actor, they're trying to promote a
to
good
promote a
actor.
good actor. Yes,
And
yes.
maybe it's just my knowledge of vaccines to know that most of them, if you're getting like a hepatitis B vaccine, you're actually trying to promote, serological antibody responses. So I don't really get the term, but
Yeah,
anyhow, putting that
I mean,
aside,
I think we're splitting hairs there.
yeah, no, I'm like I said,
Let's give them plaudits for what they're doing rather than the terminology they're using.
I know, but what I do like, so there's a couple things that are really cool about this. One, it's like a single vaccine, right? That's
Mm-hmm.
what they talk about, and that's always really the kind of the holy grail of how we want to treat people, right? If you have a disorder, it'd be great if you could just take once or annually or every 10 years or something. That's preferred. And then they're targeting regulatory T cells, and that is somewhat unprecedented in terms of vaccines. And then the way they did it is really clever. But this is good luck to them. I think it's a step in the right direction. They're treating a really, kind of. This would revolutionize the way they do it because they touch on it here. They usually sensitize you to allergens, John. Right? I'm
Sure. Oh
familiar
yeah.
with that
Yeah.
practice. Maybe I've never experienced it. Maybe you have, but they literally sit there and take a little peanut allergen and stab you in the back, and then they take a little grass and they stab you in the back, and
Well,
they.
my daughter has extensive allergy testing
Okay.
because she had that's just for testing because it's her skin would flare up with all manner of things,
Yeah, but
and so it's basically she did all the testing. Yeah, you're right. It's little pockets on your back or whatever, and so she found out. I mean, she's allergic to various man-made fibers, and then it was silver, nickel, new gold. So there's a lot of stuff
oh,
that
wow.
when she goes, for example, clothes shopping, she has to pretty much just buy cotton. Because I think we were talking, it comes up in conversation a lot because my wife will say, I found this really nice outfit, and her first question will be, well, what does it have in it in terms of materials? And then it's anything that's, I think this one that I was, a sweater I had like nylon and something else, and I was like, yep, can't touch
Yeah.
that. And then it was, yeah. So it's, and like I said, the metals is a really interesting one, and then soaps are a big one. So like
Mm,
things like polypropylene glycol and stuff that are
mm.
common in soaps, she can't touch any of those. So it's quite a challenge, So for her, with those kind of allergens, it's more of an avoidance thing. Obviously, with it being, these sort of proteins that are getting into your system, you need a different approach to that because you're not going to just avoid dust mites and things like that. And they say in this article they're actually looking at looking at food, adapting the vaccine for food allergies, and again in mice are seeing reasonable results. And the long term is that they want to go for like peanuts and cow's milk, but they want to make sure that they really have the concept nailed down before they go into the ones that are going to be more difficult to deal with. So I like, for someone who's dealing with a family member with allergies, I always like seeing these kind of stories.
Yeah.
It's I think it's true of anyone who has a family member with any medical condition. If you see something that's like means a
Absolutely.
potential advance and making their life easier, then you want to know
Yeah.
about it, and
Yeah.
you hope it goes somewhere. So
Yeah.
yeah, that's why I like this article, even though it's been sitting out there for a while.
It does look like there was a Swiss company here that had a vaccine of some sort treating celiac, right? Um,
Um, Yeah,
And so I do think there's some movement in this at the company's Anacoyan. I don't know.
yeah, Enochion, but
No,
I mean, he's yeah, it's one of the scientists in this who's a guy called Jeffrey Hubble, who's a biomedical engineer at New York University, and was one of the authors of the study. He co-founded the Swiss biotech Enochion, and again, they're talking inverse vaccines, acknowledging that you don't like the term.
don't worry about it, John. You put me in a better
for celiac disease and multiple sclerosis, so seeing this as a way to do with autoimmune disease. So again, that if that works, that's a huge step forward for those conditions. So yeah, like I say, it's once you start digging into this, there's a lot here, and if it works, then it could, like say, make a lot of people's lives a lot easier.
That's why we make such a good team, John. You kind of made me feel like, hey, I'm being petty. So thanks for that. and it is great science, and that afflicts a lot of people. The anything to do with like cow's milk and peanuts. I mean that's just it would be such a great advancement in science. But
you got to remember it's the season of goodwill to all men, even inverse vaccine researchers. You know.
Oh, John, I love it. Thanks, man.
Let's move on. Second article. This is Precise Bio completes first 3D printed human cornea transplant. So again, this is like this is never ever going to affect my day-to-day research, but hopefully I never need a corneal transplant. But this is a company called Precise Bio in North Carolina who have reported the first successful human implantation of its 3D printed cornea implant constructed of functional human eye cells cultured in a laboratory. And so they have a robotic biofabrication. Their hope is that this robotic biofabrication approach could potentially turn a single donated cornea into hundreds of lab-grown grafts to help basically relieve a need on the transplant waiting list, because it's estimated there is only one available cornea for every 70 patients who need one to see. So the co-founder of the company said this marks a turning point for regenerative ophthalmology at a moment of real hope for millions living with corneal blindness. So I'll pass it to you if you want, and I can chime in with more.
I mean, it's amazing, right?
Like, yeah.
Like 3D printers are fat. I think they're awesome. I got a little cousin or a marriage type nephew, one of my wife's side of the family. He makes all these things on 3D printers and makes a KU light and all these things. And here we got scientists who are making cornea, John. This is straight out of like a Star Trek type of science to me. And what an unbelievable story this would be in our lifetime if we're, instead of trying to get human cadavers to donate cornea, to be able to print them. This would help all of us. I think every single one of us, if we live long enough, might need something like this. I know you kind of jokingly said, I hope I don't need one, but I mean, if we can, who knows how long we might be able to live.
Oh, sure. Yeah.
Some of the advancements we've got going on, but this would be, I just think about, so my questions lie in, like you've heard me talk about this being an immune privileged kind of organ, right? The eye is an immune privileged site. It can't swell, right? If your eye swells, it's going to pop right out of your head. So it's got all these, all the epithelial cells of the eye have all these mechanisms to kill any immune cells that get in there. I don't know if you knew that. They have all these like apoptotic kind of receptors so if a cell does get in there, it immediately dies. And then it's got all these mechanisms to clear out cytokines, and the speed at which the eye moves fluid in and out of the aqueous and vitreous fluid is one of the fastest moving parts of your body. I don't know if you knew that. It pumps it at such a high rate. That's what's hard to do PK analysis in the eye.
Yeah.
Right? And so with that said, this is, I don't know. I have too much more to add other than like sign me up if this gets into the clinic at some point. If I ever, I guess.
Well, they're actually talking in the clinic because it's basically it's like the cornea can end up clouding a person's vision following injuries, infections, scarring, and other conditions. They're like say they're calling this this cornea implant PB001 is currently being tested in a single-arm phase 1 trial in Israel, which aims to enroll between 10 and 15 participants with excess fluid buildups in the cornea due to dysfunction within its inner cell layers. And they talk about the graft is capable of integrating with the patient's own tissues. So it's, I mean, it's that merge, the assimilation of the graft to obviously help in the long term. I just thought it was a pretty impressive thing to.
Yes, so you talked about near and dear to your heart with diseases. I know my brother's got one of them has a lot of eye issues, and this, in fact, I might even mention this to him and have him run it by his ophthalmologist to see if there's a potential for him here, because I know he's got some starting to get a little bit of blindness to him. But this, I'm fascinated to know what the cell line they used, how some of that integration you talked about, what they're doing to kind of prevent any inflammation. It's wonderful stuff. And Brad, if you got a comment for us, please.
No,
go
Dom, I
ahead.
was just, I appreciate. No, gentlemen, it's fascinating in a sense. It's merging both articles together, talking about basically priming the immune system to prevent any kind of graft versus host disease as we start navigating this area. Not just look at simple cell structures, but look more at tissues or specifically organs. If we could 3D print those, and then develop a tolerance so that we don't necessarily have to give people continuous drugs to suppress the immune system. I think we're merging both of these articles in a way that will fundamentally transform the way we treat people moving forward. It's exciting.
No, that is a great point, Brad. That would be a fascinating thing. Whole organs with some sort of, what did they put on the other article? I think they put a protein on there to prevent it from being immunoreactive, to make it more tolerant. That is a great point you just made, Brad, and really cool. John, any other thoughts there?
Nope, not at all. I just, like I said, just love seeing these things, and you sometimes you see these and it's like, we have to talk about that, and that's an easy one.
It's really cool. And again, it's really, to me, it makes me feel like I'm watching Star Wars or something, or Star Trek more. With just some of the medical advancements here, but okay, John, we got one last one.
One big one.
It's a good cheery one. I won't hate on this.
And this is again, this is one that has been out there for a while, but it's always, I mean, corporate companies are all about making money, etc. But it is nice when they do something that helps a population, however small. And this is actually talk about Mattel, and the headline was Insulet Medtronic laud debut of Barbie with type 1 diabetes. So basically, as anyone who's grown up with or has kids, you see the evolution of Barbie over the years, and I've had my daughter went through her Barbie phase briefly, and then obviously moved on, and the Barbies are now shoved away in the attic. But there's a change over the years. My wife had like Barbie, and the British rival was Cindy, so she's got a soft spot for those. But this was actually announced back in July, and Mattel said that they were going to have a Barbie that represented the first version of the doll depicted as having type 1 diabetes. So this Barbie was developed in partnership with the research and advocacy non-profit Breakthrough T1D. She comes with an insulin pump attached to her waist and continuous glucose monitor on her upper arm, held in place with a heart-shaped medical tape in her signature pink. She carries a small purse that can hold a smartphone equipped with a CGM app, plus any additional medical supplies or snacks per Mattel. Finally, Barbie's matching top and skirt are covered in light blue polka dots, a reference to the blue circle that has served as universal symbol of diabetes awareness since its introduction by the International Diabetes Federation in 2006. What I like about this is the doll is part of Mattel's Barbie Fashionistas line, created to highlight diversity in skin tones, body types, disabilities, and more, which also includes Barbies with blindness, hearing loss, and Down syndrome, amongst others. And it's basically it's showing kids that they're not to be isolated. When it's part of helping their acceptance with their peers, etc. And I think it's great from that perspective. I'll pass it to you, and then we can chat more.
Yeah, no, I agree with especially that last point about like just if you make a child aware of something, they won't tease it, right?
Yeah, exactly.
That's kind of
yes,
it. So that if kids are like, all the things you mentioned from Down syndromes to type 1 diabetes, just understanding, even if you're a healthy child, having these things and seeing it, and then when you meet somebody, you don't think there's something wrong with them and you want to tease them, right? It's kind of, I don't know if normalizing it's the word, but it's awareness and compassion and empathy and all those great things. So it's a wonderful, they should be applauded for this. I think it's great and then the attention to detail is just cracking me up.
Sure. Yeah. That's why I had to go through it, what it all is. Yeah. It's brilliant.
Yeah.
And that's what the headline talks about, Medtronic and Insulet, who make the technologies that these type 1 did the pumps, etc. And they both said that a Medtronic spokesperson said that no manufacturers were involved in Doll's development, but in a post on LinkedIn, Medtronic's diabetes, Medtronic Diabetes shared the launch news and congratulated Mattel and Breakthrough T1D on the doll's debut. And it's sort of saying that because of the way they've done the research, it totally mimics how the pump sits on a patient and things like that. So it's not just doing it, but it's the attention to detail is also a key part of it. And I think it's great. And Brad, I see you have something to say.
Yeah, John, I was just going to echo. I think, Dom, you're spot on. It makes kids a lot more adaptable to what were initially perceived as issues or challenges, but it's just commonplace of life. I was just going to point one thing that's kind of exciting that really just broke this fall and came out of early discoveries in the New England Journal of Medicine in 2023, looking at preserving beta cell function with patients treated with baricitinib for 48 weeks. So there's 2 trials actually ongoing right now with baricitinib that could potentially delay the onset of type 1D or potentially eliminate it altogether.
Wow.
I think it's going to be interesting to see where this comes. There would be nothing more exciting than to have this, God bless her, this short-lived Barbie that comes in with the pump and comes out with the pump, because we basically found a cure. So let's hold on tight and see where that data comes.
Yeah, that is really cool. I have a niece who's got type 1 diabetes, and I just wrote a note. I might try to send her one of these dolls here for maybe the new year. I don't think I can get it to her by Christmas, but I think it would be a great little gift. And as for some sort of like vaccines it gets back to like COVID. I think the vaccine world has just exploded. I do think over time we might have a vaccine to cure just about everything, and really for especially any sort of autoimmune diseases are challenging. Allergies, immunology, those types of diseases are most of the time. Most of the therapies are just to mitigate the symptoms of it rather than curing it. Where vaccines could surely offer a cure. And Brad, if there's that, I might have to research that a little bit more. But so I guess it's only if it's to prevent it. I see. It's not necessarily something for an adult. It would be more for treating children, but still wonderful. It's only a matter of time. If they can do it in a child, then they'll engineer it in a way that it might be able to help adults. But what a great story there, John. Look, you lifted my spirits nicely as we go our agenda here. Yeah. I don't know if you have any last parting thoughts on that one, but
no, I like to say it's like, for me it comes down to it's like you say about not highlighting the differences, or getting kids to be more accepting of others, which I think is a big deal. But we know that growing up, kids can be cruel. So anything that, like you say, that takes that picking on someone because that's slightly different is a good thing in my mind. It's
nice touch with the polka dot clad, the blue signs of the insulin of diabetes type 1 support, and you can get an accessory, is what they say here, to dress the Barbie in support of that as well. So just good job all around. Applauding Medtronic and Insulet.
Yep.
Great job. Yeah, really cool. Well, with that,
let's move over to our main topic. I'll get things started because, as our special guest is very aware, this is a topic that I will sit and listen and learn rather than driving too much of it. We find that the LCMS topics are the ones where I'm intimately integrated, and then there's other things like this where it's we're into black box territory for me. So it's listening and learning time. Anyway, we have a special guest. Brad Nawa, West Region Director of Alamar Biosciences, to talk about NULISA, a new ultra-sensitivity approach for protein quantitation. Welcome to the podcast, Brad. Like I say, it's largely going to be a discussion with you and Dominic. But I'll chime in wherever I can, because I've heard a couple of presentations on this, and I know we have an instrument that we're getting up and going in Lyon and starting to show capabilities. So I think for this topic, let's at least start. First of all, Brad, give you a chance to introduce yourself, talk about your current role and your previous history, and we'll then take the conversation from there.
Excellent. John, Dom, thanks so much for the invitation. Excited to be here. Just to provide a quick snapshot of my career, came out of a HEMONC lab in grad school, was on a Hughes grant and decided to get my MBA. And at that time, I started entering into the commercial space. I've worked for a variety of different companies, different platforms, specifically around proteomics, flow cytometry, electrochemiluminescence, transitioned over to proximity extension assay technologies, all for small upstarts at the time that were just under 50 people and then starting to scale. So it was a very exciting time to be in this space, still is. And had the good fortune of joining Alamar just as we were ready to commercialize the NULISA and the ARGO platform, which runs the NULISA chemistry. And that was in January of 2024. And I can tell you, it's been a whirlwind for us as a company, for us as a scientific community, as we're starting to make these advancements. It's been extremely exciting. We're proud to announce, actually, as of this week, we celebrated our 100th publication. That's less than 2 years. And we will have deployed over 100 instruments worldwide on the
Nice.
ARGO platform. So it's been quite exciting for us as a company, but more so exciting for science in general. And I say that because we're able to detect proteins now in blood that we haven't been able to detect before at extremely low levels. And that's incredibly important as we start progressing the clinical components of disease and looking at the etiologies and how to better treat them. Yeah.
And Brad, before we jump in, I gotta say to our audience, I believe the first time I met you was in 2006. Does that sound about right to you? And that was
you are correct, sir.
I mean, that's gone by real. Like as we enter 2026, that's like 20 years, Brad. You've been having to deal with the likes of me, and I still remember that the first, there was the electrochemiluminescence system where we met. And Brad, it was a pleasure working with you and helping us to really bring that platform into the forefront, because that was still in its early stages. And so when we acquired the NULISA system, and we acquired it in Lyon, I knew we had interacted with some people at Alamar, and then lo and behold, you kind of rang my doorbell maybe a year ago, and when I found that out, boy, I thought, man, Alamar's lucky to have you. I think it's been, someone like yourself. To me, it really validated the system a little bit to have somebody such as yourself that, Brad, I'm gonna give you a nice applaud here. You wouldn't have joined this company if you didn't believe in it. I think we talked about that when you first joined. You really do have a good sense of how these things operate. It really made me feel like, oh wow, if somebody who has decades of experience in let's say biomarker platforms or LBA platforms believes in this system, well, that removed a lot of any of my if you can't tell, I'm sometimes a little skeptical.
Sure.
But Brad, maybe I'll let you go. But I just wanted to make sure, just to know that it's been a pleasure over the last 20 years working with you, and I'm looking forward to another 20. Well, maybe not that many, but yeah, go ahead.
Yeah,
Brad.
I mean, as an outsider, I know that we've seen talk of a few ultra sensitive platforms like this that we've brought in, but really haven't translated to commercially supporting much on actual projects. So we still tend to stick to and I know I'm talking about a department I don't work much with, but I don't hear any mention of those platforms other than we have them. So we tended to stick with our workhorses. So it's exciting to have one that may have some potential to take us into this realm.
Well, before Brad starts, you make a good point, Brad. John, they didn't work really as advertised. They had some successes to get ultra low, nothing near the level of NULISA, and Brad's going to share with us just how sensitive this can get.
Mm,
But it still had a lot of dependency upon the antibody you were using. So if
sure.
you were using that ultra sensitive platform, if you could get your hands on that antibody, it worked on almost any platform. So with that, right, Brad, I feel like that's where. So it was some of it was just marketing. Again, there were some successes, but it was always more the antibody. I feel like the NULISA system, the antibody's still important, Brad, right? And maybe I'll let you
It
tell
is.
us. Why don't you just dive in
Yeah.
and
No,
tell us
Tom,
tell
it's
the audience a little bit more about the platform itself, because John and I already have some knowledge of it. But yeah, go ahead, Brad.
yeah, no, so no, it's a great point, gentlemen. And really it is content driven, right? You're only as good as the antibodies that you're using to detect the proteins that you're trying to pan for. So we are a ligand based assay platform in the sense where we bring a sandwich, basically two antibodies in proximity to a protein. What's different though from our strategy as opposed to other technologies is we don't work on just amplifying signal, but we work on reducing background. Um, and there's actually a beautiful cartoon on our website that demonstrates NULISA and its entity, and it's basically two antibodies that are coupled to two genomic sequences that identify the protein of interest. What we do instead is we use two capture approaches. We polyadenylate one and we biotinylate the other. And what we have actually do is purify that specific immune complex that we've formed, and we can effectively scale that from one target to potentially 200 to almost over 1,000 targets if we really ideally wanted to, and be able to take that technology and move that forward. Just to so the biggest difference between us and other methodologies is that we can really purify that immune complex before we go for that amplification step to detect those proteins of interest. I'd say the other thing that was missing really in the marketplace for the longest time was something that was fully automated, something that got rid of potentially interoperator or scientific interoperator variability, which could count from anything from 5% to 30%, depending upon the operator and the time of day the assay was particularly run. So the Argo itself is a self-contained platform that runs the NULISA suite of assays. So what we've effectively done, if you can work a microwave, you can work this Argo. We're trying to de-risk as much as we possibly can an assay from its inception all the way into running into clinical practice. We're trying to make it more like a blood analyzer. And that's really where the vision of the company was, is can we effectively go from early discovery all the way to clinic with these potential biomarkers? Can we plug and play these biomarkers as well? And the way the chemistry is set up, we can absolutely do both of those.
That's a great point, Brad. So you're not only are you making it's just a more robust platform because it's a little more automated, right, or more completely automated from what you're saying. But and then tell us a little bit more about the. I think you said there were like, only are you putting nucleic acids on as unique identifiers? Do I have that right on each of the
Yeah,
capture
John.
antibodies?
So what we've done is we've linked nucleic acids that come into proximity. This ligation event actually creates its own code, if you will, or barcode that identifies the protein of interest. So these will be unique sequences that are attached to these specific monoclonal antibodies. We predominantly use monoclonal antibodies directed towards a potential target. So for instance, IL-6 will have its own unique sequence. We then amplify that sequence, not too dissimilar from proximity extension assay. The difference is we use ligation events, so two blunt ends coming together, which allows us to really plug and play this content a little bit easier, as opposed to proximity extension, which the affinity of two overlapping sequences, there's different affinities based on the A, T, C, and G contents. So really, we work independently. Each assay works independently, but can be scaled appropriately as well. So we can add as many of these targets as we'd like into the soup, if you will. We don't get a lot of cross reactivity, if any, only because we do work predominantly with monoclonals. We try to control that as much as possible with monoclonals, and we try to look for the best targets or antibodies that are possible out there in the commercial space. So there's a lot of engineering that goes in on the front end of these assays before we actually release them. But the point is it's really that double capture step coupled with that barcode sequence that gives us an idea of exactly what that protein is and where the relative amounts of that protein is, specifically in the serum, the plasma, or the CSF, or a lot of different biological fluids. And what that does really is informs us to a much greater extent of what's going on within that proteome space, specifically relative to the blood, and trying to mimic what we can find in the CSF. So for neurodegenerative diseases, for instance, we can really be able to effectively track those changes, whether they're either on therapy or looking at the early progression of diseases, and we could potentially infer some pretty
unique, right? Or in
yeah. I'm sorry, guys. I'm
just
looking
like
for a word here. Yeah,
a never before seen ability to kind of monitor, and we're going to get into like the application of the system, but it has utility as a biomarker platform, a diagnostic platform, a PK platform, right? But before we move to that, right now you guys have like a 256 plex, right? That's the most analytes. But if I understand you right, you could actually go much higher, right? That you have you have an infinite number of nucleic acid sequences, that barcode you're talking about. So this is like the sky the limit, or is there a cap to the number of like analytes you could do in a single shot?
Yeah, Don, I mean, I don't want to necessarily say we have a limited number of sequences that we could use, but understand those are in the thousands. So we have the ability to scale up.
I don't think anybody needs that many just yet, right? Like 256 is still, and you get like 90% hit rate in a CSF sample on that, right? It's something, it's really off the charts. Yeah. So with that, maybe it's a nice segue into our next topic. Tell us a little bit more about the application of the system.
Sure. Just to touch back on the relevancy. So you're exactly right. So typically in healthy normals, what we try to do is benchmark ourselves and see, can we find a protein signature in healthy normal plasma and serum or CSF? And we usually get anywhere from 96 to 98% sensitivity on these targets in the plasma and the serum. That's one of the hallmarks of the technology. So if we can establish these baselines and watch the progression of disease, or if we're comparing a healthy cohort in a phase 1 trial up to a treated cohort, we can actually understand how far we can either knock down that signal or see how far that signal amplifies from baseline. So we can really infer some ideas of whether or not we have an MOA, or the target's actually engaging what it's supposed to do, or we're seeing the cascade effect downstream as we look. The content is driven, back to this point though, the content is driven for biologically relevant materials. So those 250 have been highly curated. And what we try to do is really understand biologically what we can do as far as these specific targets go. So you've got small abundance of IL-1 beta, et cetera, and small IL-4, for instance, that we're able to detect in this healthy normal population, really be able to track effectively. Back to your point though, specifically on applications. So we have the ability, actually, the platform is open, so you can design your own assays. You can use your own proprietary content or antibodies to drive the solution. PK, pharmacokinetic, pharmacodynamic assays, we seem to excel at. This is not too dissimilar to some of the work that KCAS has been working on with regards to PK/PD biomarkers, to be able to track those efficiently or effectively. So it's an open platform. A lot of it for patient stratification, understanding early disease diagnosis, or potentially not in a diagnostic fashion, mind you, but just understanding right now we're RUO, but understanding exactly when early onset of diseases and trying to infer some ideas from pathways of what exactly might be happening as we start developing. Then if you intervene with a therapeutic on the trial, you can actually see how that proteins are changing after administration.
Yeah, it's fascinating, Brad. And then I did have a quick question here. So can you still analyze tissue samples on this platform? I believe you can, but
we can. Yeah.
You can,
Don,
right?
We can. Yes, sir.
Yeah.
We actually set the platform up. We've optimized it around serum, plasma, and CSF, but we've used a variety of different matrices, including tissue lysates, brain homogenates. We can use vitreous humor, back to the eye question.
Mm-hmm.
We use skin tapes, dry blood spots, and dry plasma spots. We seem to be particularly good at being able to detect these. So as we start democratizing these trials and maybe more at-home testing kits or at-home monitoring kits, we'll be able to address that market. Again, just because of that dual capture, we're able to get that level of sensitivity. And we get a pretty consistent signal. But we're working right now with a lot of DBS or DPS platforms, and working with a lot of the research community, so we can better understand exactly how the platform performs and really guide our clients to what the best solution is. So I would say we're really a group of scientists that just so happen to have different roles, whether it be finance, commercial, and discovery. But the idea here is can we bring real value to the scientific community?
Yeah, I think it's important to emphasize it can be used in almost any matrix, and we're very optimistic here at KCAS Bio with some of what we're seeing with the system. Um, yeah, but I do want the audience to emphasize that it's not limited to just like a plasma serum or CSF. And did have one follow up question around the um, the utility of the system, which it's escaping me right now. It'll pop back into my head, Brad. But maybe you can continue on with a little bit more about. I think we did touch on the applications.
Yeah, what about some of the therapeutic areas that you're seeing utility? 'Cause I mean, I know that like we have like some of the CNS biomarkers. You talk about the ultra sensitive ones for Alzheimer's, etc. I mean, I assume that's a key piece for you, and what else for you? Where else do you see? Where's the areas that this is really your sweet spots?
Sure.
Is the question.
Yeah, John, it's a great question, and actually, it was somewhat serendipity. And again, you always have conversations, and you have these connections, and it leads to something. You just got to be willing to keep pulling through. We initially launched a 200-plex inflammation panel as a me too into the market. Let's look. We can measure these inflammatory cytokines, and then we engaged with some of the neurodegenerative leaders within the space, and they had come to us and said, "Hey, we see where the technology can go as far as the sensitivity and the merits of being able to scale that. We're really trying to understand what's happening in that CSF compartment." So for CNS neurodegenerative diseases, we really can't just take spinal taps. We'd like to understand, can we get through that blood-brain barrier and understand what the compartment looks like? And we've been able to effectively look at small peptides, BD tau, for instance, the phosphorylated tau 217, the brain-derived. We just launched that product actually a few weeks ago. And we had some great presentations at CTAD, and most recently, our 100th publication coming out of the University of Singapore that really provided some really good insight into brain-derived tau and the merits of the NULISA platform relative to what other technologies are out there. What we're looking for is really good sensitivity to diagnosis. Right now, it's using PET imaging to get a good idea of exactly whether or not you do have amyloid and exactly what levels of you may have relative to Alzheimer's disease, but we know there's a number of different co-pathologies with that. So with the CNS disease panel, the 120 targets that we have, that basically gives us some good insight as to what other synuclein pathologies, as well as looking at TDP43 pathologies. We can also look at the blood-brain barrier and understand what's happening within that membrane. There's a lot of different things we can take on these larger panels that just give us a much better snapshot of time of what's happening within that CSF compartment or that blood compartment, so we can really understand the pathology of the disease. Because the key here is early detection, right? Before the pathological changes start to manifest, can we look at these biomarker signatures to get a good idea? And that's really where the mission is we want to power the precision proteomics for the earliest detection of the disease. So back to your question, we've really hit the CNS market, the neurodegenerative market. We're now moving more into the autoimmune or the immunology market with our immune function panel that also has some great cancer targets on it, PD-L1. There's a lot of, it's more of a Swiss Army knife, but what's going to be exciting about us is we're going to be moving into the cardiometabolic space. We're going to start building out this content more and more strategically as we start moving it forward. We're fortunate enough to recently be aligned with the PPMI, with the Michael J. Fox Foundation, directed to create some new targets for them for their initiatives. So it'd be nice to see. I tell a lot of my colleagues or my friends that I'm hoping that we can get a ring fence neurodegenerative disease over the next 10 or 15 years, but technologies like Alamar that'll give us a lot more insight and a lot more opportunities to solve these problems.
Yeah, sorry, John. I got a couple things here. Brad, you mentioned dried blood spots. I know the microsampling by Mitra and Tasso devices are of interest, and that's something we may explore as well. We've got something we might be working on. Has anybody actually done those kind of microsampling matrices?
Yeah, Dom, we have, and we've been quite successful. We've gotten better part on depending on the panel. So a lot of times what we'll do is we run a technology access program for early access to do some testing for someone that may not be enabled right now with an ARGO or access to NULISA, or we'll recommend them to CROs like yourself to be able to run some of those tests. But so far we've run about 8 different dry blood spot or dry plasma spot sampling devices. We've gotten what I'd say very impressive results, greater than 90% recovery of the vast majority of these targets. And we're learning more and more. What we're learning too is how these proteins actually behave, whether depending upon what they're actually being put on as far as a fabric, if you will, or a bed where these proteins are actually lying, and what the best extraction processes are as well to be able to get a robust protein signature.
Yeah, I'd like to learn, but maybe I'll take that offline. But there's the extraction process itself that dilutes that sample a little bit.
Mhm.
It's incredible that you're getting detection levels from those types of samples. That's really
And can I ask a question? I mean, like I say, obviously, I've never come close to I haven't even seen the instrument. But what kind of sample aliquots are you talking for this?
Sure. So, John, typically we require the ARGO runs with 25 microliters
Yeah.
of sample. The
Yep.
assay only requires 10. It's automation, so there's 15 microliters of dead volume. That's
Mm-hmm.
for our multiplex. Our singleplex are a little bit higher, 35 microliters of assay. It's just the way it's set up. And then for our dry blood spots, it depends on the extraction process and what we're able to provide. We effectively dilute that as
Yep.
we run the sample anyway, roughly about 1 to 5, but we run neat. So we don't need a pre-dilution because we're able to effectively grab about around 12 logs in aggregate of dynamic range across the entire panel, 5 to 7 logs of dynamic range for any particular target in that panel.
wow.
So can I ask a further question? So obviously, with dry blood spot that I've worked on with small molecule, the first steps get the drug off the dry blood spot, and for us it's usually methanol. We take it from there. I assume that's going to be too harsh to your antibodies, etc. So what are you reconstituting in to get it off the dry blood spot to then go into the system?
Yeah. So as far as the extraction processes are concerned, there's a variety of different buffers and solutions that we use in order to extract that. Just to save a little bit of time here, we won't get involved in the nuances, but it seems to be a little bit different based on each platform and what actually works best. I would say, and we empirically derive that. What we're trying to do is find out what the best platform is for the potential utility of the marker itself or the potential of the disease itself as well. One of the fun things we've been able to do is use this platform to basically create a proteomics clock of aging. There's some good data that we've been presented, the biomarkers of aging, and we've been working with a lot of different groups to potentially use DBS or DPS as a way of being able to monitor age or molecular aging relative to protein signatures to understand what's going on. So I'm sorry to go off on that tangent. It's just something
It's good.
of where it's actually going. Go ahead, sir.
Yeah, and then you even touched on like the Swiss Army knife. I kind of jotted a note down. It seems like you're actually working towards like having a diagnostic for like general health, right? Is that on your where like it doesn't matter your age, even if you're feeling good, or sometimes people they just don't feel great, and doctors have to throw a differential diagnosis at it, and they're throwing the kitchen sink at it. You might be able to at Alamar come up with something that gets a more I would say a faster or maybe a more simplified version of saying, hey, it could be here because of these elevated markers. Is that kind of the vision, or do
Dominic, I think you're really onto something relative to where the diagnostic community tells us where to go. We're listening, but for routine monitoring and really understanding the pathology of disease or understanding what's going on, it's going to take a lot of data, right?
Yeah.
We're working a lot with biobanks now on retrospective samples within clinical trials to better understand what's going on with relative to the proteome there. But we're making some significant strides on the data sets and what we're being able to pull out And I agree with you. I think we're working with a handful of DTC or direct to consumer companies right now to really understand things as simple as, hey, I've got mild cognitive impairment. Do I potentially have early Alzheimer's pathology? Should I get this tested in reflex? And you can develop these as LDTs and just make some suggestions and put them in touch with the neurologist. But I think the sky's the limit of what we could potentially do as we start analyzing these proteins and making and inferring these patterns. Because it's not going to be potentially one protein that anchors us into a potential disease or a solution, but a litany of these proteins. And then being able to substratify a lot of these diseases now, we're finding more and more of these subtypes or endotypes of these potential
Mm,
diseases or these co-pathologies. I mean, we know this well with lupus. We know there's like
mm,
4 or 5 different endotypes. We know also with people that come in with dementia, they may have an Alzheimer's profile. They may also have a Parkinson's-like profile. They
yeah.
may have a Lewy body dementia or frontal temporal dementia. So we're really trying to make strides into understanding. And as these therapies start coming out, and for different targets and different methods, it's going to be critical that we really understand the pathologies of diseases to basically administer the right therapy.
Yeah, now you got me really, my head's like swimming with like thoughts on this, but maybe we'll move on because like even like gut health can impact some of that stuff too. And you might be able, or cardiac health, metabolic health, all those things. You might be able to, like you say, stratify in such a way that it's like, oh, this person might be having this symptom, but here's the causes over here because we've got all this information. Great stuff, Brad. We've got a little bit of time still. I don't know if we want to jump into like some of the advantages of the system. Maybe that seems to be on our agenda here. You want to talk to
Certainly.
that, Brad?
Yeah, Dom, I'm happy to. And I'd say the biggest thing is our sensitivity, the attomolar levels. The fact of, when you're running any kind of immunoassays, you want consistency across platforms. You want something, for instance, if I'm at a pharmaceutical company and I develop an assay and want to port that over to a CRO or a trusted partner that can run that assay, I need to make sure that we mitigate as much of the as much potential mistakes as possible or errors that could occur as you run these assays. The fact that these things are fully automated gives a lot of reliance or at least relief from the researcher's perspective that we should see indiscernible data. And when we leave these instruments, when we actually launch these instruments, we make sure we test that as well. And we see very little differences in the performances from Argo to Argo. So I think that's one of the biggest advantages as the Argo platform as a whole. The NULISA, we talked about the scalability and the levels of sensitivity as far as the differences are concerned. But I think what really wows people is when they're able to detect markers, biomarkers that they didn't think they could detect before, or they're finding new discoveries that before were just commonplace or maybe dismissed only because the methods weren't able to detect that level of sensitivity. But we're only going to be as good as the research that comes off of discoveries like mass spec, for instance, or the development of new content for these new targets. These post-translational modification of these proteins are going to be important too. And we're going to build that content to drive those solutions as well, because it's good. It's a big open field. It's a very exciting time, I guess, is my point, to be in this field.
Yeah, no, there's so much to unpack with just I'd often say to people who are doing like, kind of discovery proteomics, discovery biomarker, they might want to do a 10-plex or even a Luminex panel upwards of 30. And I'd say, hey, just don't want you to invest in something and get a bunch of BQLs because good chunk of what you're going to see is below our limits of detection. That changes and is almost revolutionized with the Alamar system, right? That becomes a non-stopper that you're going to get data. It's just, I think there's still a little bit to be vetted out in terms of what it all means, right, Brad? Like that is really, to me, the biggest kind of unknown yet is if I do have alamolar levels of a given biomarker, maybe outside of CNS, because I think those are clear, a little more vetted out. But what exactly does that mean? But it's really you touched on a lot of things. The thought of being able to have a system here in the U.S., a system in Lyon, and you're saying the robustness of the platform we're going to get within like what 5-10% same
Sure.
numbers. Is that what you're saying? So that really does kind of level the playing field a little bit in these ultra sensitive detection levels. and really does make it a potentially the future of biomarker analysis, right? And it's still it make it I think I don't know if you have enough information around, hey, if you send the same, if you send me 3 lots of a plex, we test all 3 of those lots, we're going to get reproducible, robust responses. A satellite lab would have the similar responses. Has that type of stuff been done, kind of vetted out just yet, or maybe?
It has, Dom.
Okay.
Yeah, it has. So we've done some concordance studies with working amongst different labs. We ourselves have multiple ARGOs at our Fremont, California site, so we can effectively test each of those independent ARGOs as its own separate assay, running those controls or running the same set of samples, so we know how those antibodies or that platform differs. Before we launch a platform, we do look at the reproducibility from lot to lot before we actually release that assay. So we want to have control of that assay. That's not to say that as we expand those platforms, explain the content, there'll be changes, of course, because if you start adding more content to this, the suite of assays will change. But the point is that we do the rigor on the front end to make sure we have as close to a diagnostic level of quality in an RUO market. So there's a lot of research and homework that goes in with the ISO certifications, et cetera, that we do, so that we are ready to bring out the most robust suite of assays, and the most that have reproducibility specifically in your hands, because it doesn't do the researcher any good if they find something and they can't replicate that in the same
Yeah.
sample set.
Yeah,
It
exactly.
becomes challenging.
Yeah, Brad, I want to emphasize the utility of the instrument real quickly here. You've got these biomarker panels that we're talking about, but a lot of it is I call it the home brew. Maybe you call it a different thing, where it's really a single analyte you're looking at, and we've seen some nice success with taking biomarkers that were developed to be ultra sensitive on other platforms, ECL, maybe even the Samoa and things like that, and taking that and flipping it over to your instrument and seeing, log scale, better sensitivity. That's so you can do multiplexing for biomarkers, you can do homebrew or single plexing for biomarkers, and then really it could revolutionize the PK space a little bit. And I don't know the utility as much there, because sometimes you just don't need that level of sensitivity for PK
Yeah.
assays. But certainly
Yeah.
in pediatric studies and ocular fluids, that this could be a real
Mhm.
advantage there. But is there any? Those are kind of, and then you got this, like, it is almost like a diagnostic or a CLIA tool as well. Are those like the 4 or 5 real areas that you all are trying to grow, or did I capture them all, or is there any more that
Yeah.
you all are working on?
Some of these assays are good enough. They're fit for purpose. Do we need the sensitivity? Perhaps not. If we're working with precious matrices, or we're looking at new ways of extracting these proteins and we need that level of sensitivity, then really NULISA is the optimal choice to at least take a first pass and see what we can discover. I think I would also point out is that the chemistry itself is, we're really geared towards proteins, or using antibodies to detect these proteins. We can also conjugate peptides to this chemistry, this NULISA chemistry, so we could effectively profile autoantibodies if we have specific autoantibody needs that we want to look at. We could also look at protein-protein interactions as well. So there's a lot of different utilities we can use with the NULISA chemistry that just aren't to simply understanding soluble or membrane-bound proteins. So I think the sky's the limit of where this technology and the platform can take us.
Yeah, no, it really, you got me thinking a lot about like autoantibodies, protein interactions, what we get asked often about, that can be somewhat challenging if you don't know what it's binding to, and this seems to maybe have some ability to do it. John, we've covered. I think we've covered most of our agenda items here in terms of advantages and maybe the future of ELISA. But I don't know if you had anything to add, because Brad, I think you kind of, maybe that is the last kind of topic we can touch on, is just the future of the NULISA platform. And I think you kind of shared there, but any other thoughts on where you see this going? I think you might have
Yeah.
touched on it, but
Yeah, I think that makes sense. And then we that will wrap things up nicely, I would have said.
Okay. No, Dom, I would just add the simple fact that you're only as good as the data. As we start migrating towards artificial intelligence and being able to pull data sets together, doing the meta-analysis on these different trials, we're going to need good, reproducible, robust signatures and proteins and data to pull from. I think technologies like NULISA, or specifically NULISA and Alamar, are going to lead the way in what we could potentially do. I just add that we've worked in with the Gates Foundation as well to provide these kits or these content to investigators around the world, so we can have an open source database where you can actually start pulling some of these information specifically around these neurodegenerative diseases. So I think that co-sharing of good data is going to be immeasurable to what we will be able to accomplish.
Yeah, that gets me to maybe another question around the data itself. Are you guys using things like omics? Because you're generating a ton of data. How are you managing the data? Is that, because that's probably your next kind of, let's call it, mountain to climb, right? Do you have a way of, because you can't just use Excel files, right? What are you using to kind of assess this data?
Yeah, we have our own proprietary software that we've developed. But it's amenable to any external software as well. We're cloud based because there is immense amount of data that comes off this. I would say what's coming out in the future is hang tight, because we're going to do some pathway analysis as well from our next suite of software. So it's really going to give you some very good insight as to what's happening relative to pathways. You can look at 250 markers, your 5 or 10 favorites, but you're not really sure what goes on in the other 230, 240. What we're trying to do is simplify that, so you have a good idea of potentially what else may be happening ancillary, so you can potentially infer some new discoveries.
Yeah, and that's great to hear that you're. What about the actual storage of the data? Have you found, because that's become a little bit of a challenge with like high res mass spec or even some spectral flow cytometry. Storing that data for a decade can be, for lack of a better word, expensive. Is that something you guys are running into as well? Is it like, I know you're generating, or is it not quite the gigabytes with the data?
Yeah, it's not quite to that level yet. We still can transfer the data because we're still working in fairly manageable panels. The data size is not so difficult. Direct downloads, it depends on the speed of the access from the ARGO. But yeah, you're right. It's something we're going to be concerned with as we start scaling out these assays. But it's one that I'm sure that the team internally, our engineers,
Yeah.
will certainly find a solution.
Yeah, it doesn't seem like it's as much as like a spectral
No.
sample or even a high res. That's like John, help
Yeah,
me.
the full scan high res is a nightmare in terms of the amount of data it generates. So, yeah, it doesn't sound like that yet.
Yeah, Brad, any last thoughts before we put you on the hot seat?
No, Dom, I really appreciate it, John, for both the invitation today and to highlight what NULISA could do. I would say hang tight. There's a lot of exciting things coming out of Alamar, and it's great to be partnered with gentlemen such as yourself to help propel this and propel the science. But thank you.
Yeah, no, I'll just add we're gaining a lot of traction, a lot of excitement. I'm sure we'll be interacting more in 26, and with a bit of luck, Brad, we'll get one here in the U.S. I know that's on our thought process. We just want to make sure we get more utility out of it in Lyon before we bring it here. But I wouldn't be surprised if we have one in the U.S. sometime in the next year or two. So, all right, Brad, we're gonna put you in the hot seat. I hope you're ready, Brad. I'll go with the first question to you, Brad. What's your favorite adult beverage?
Okay, guys, you're going to kind of call me a bit of a Sally here, but
I could have predicted this, Brad. I should have said you don't even drink, right?
No. Truth be told, Dom, I do.
Okay.
It's a grapefruit Hefeweizen. It's by a company called Schurferhofer. Schurferhofer. I am trying to do my best German. I apologize, but it is a grapefruit Hefeweizen. It's got a carbonated grapefruit. It's extremely good. I discovered this at Epcot in Walt Disney World. Found it in Germany, which was the little country there in Epcot, and it goes down just like water. It's absolutely delicious. So I would recommend anybody try it. It's not incredibly cheap. It's about $2.50 a bottle. Not incredibly expensive either, but relative to a Busch Light or something of that nature, Bud Light, it's not in the Not in the same
It's not a Bud Light audience, Brad. I can tell
exactly.
you that.
Okay, fair enough.
You're certainly not on this
No,
podcast.
it's a bourbon whiskey audience. No,
Okay, easy
maybe
enough.
not. Gin and tonics and things like that. But no, that's great. Have you tried the Sculpin grapefruit beer there out in California? The Sculpin Brewery in San Diego. They got a
I
great
have.
grapefruit.
I have. As a matter of fact, yeah, I've got a San Diego Surf Company T-shirt on right now. I have. It's very good as well.
Yeah,
I would
but that
highly
was so.
recommend.
I'm going to try to track that down. My wife loves the grapefruits, and anytime I can get a little treat for her, it's a win. So, thanks for that. And yeah, no, I think maybe you're one of our first beer drinkers to come on and throw something down like that. But you got me thirsty for wanting to try one of them. I bet you it goes down like lemonade almost, or grapefruit juice,
It
right?
does. It
Yeah,
does.
That's dangerous, isn't
yeah,
it?
very. All right, John, you want to ask the next one, or
Yeah, let's go with a favourite dish to make. It's
Oh,
always enlightening.
no, that's great, John. Thank you, and it's an easy one. I'm a meat guy, 100% carnivore. So a good steak on the grill is absolutely my vice. The grill with broccoli.
okay?
Not a big potatoes guy, but we'll just keep it meat and vegetables and keep it simple.
Yeah, and like, so is it a fillet? You like some sort of ribeye, and do you put a little salt and pepper on it? What do you do? Or do you
Yeah,
just
yeah.
like,
We basically just salt it, Dom. My wife is pretty much our chef. In fact, she's forbid me from going into to using the grill. I mean, I will do everything else. Keep in mind, I cut wood, do all that such. But for grilling, she says, "Hey, I'm going to grill, but we salt the meats.
okay?
Predominantly, that's all we use, and just use the natural flavors to.
And I'm all into the salts these days. Do you use some fancy salt? I got these flakes. It's from like Medford salts or something like that I really like. Or is it just whatever we got in the cabinet we salt it with?
She has a proprietary salt. I've not really identified it. I mean, it's not, when I say proprietary, and I can't exactly. I'm not sure. I do know it's in a special bottle. It's the same thing she's always used, so we're pretty
Yeah.
consistent.
So,
Um,
so, so on the salt front, we go through moulding at a hell of a rate here. But I was so obviously being someone from the UK based in the US, I does a lot of sites of people who sell British food. Some they produce over here with different cures and stuff, and some are just selling commercially things that are produced in the UK. And if there's one called Scottish Gourmet where I'll get my Scottish sausages, bacon, and things, but they sell a salt that is blackthorn filtered, and it's actually fascinating when it's like they have these big filters set up on the seashore in Scotland, and they run the water through this blackthorn's just a shrub, and filter it that way. I have the salt. I haven't tried it, but yeah, I just love getting
Yeah.
sucked into these sort of foodie things where there's a different producer saying we get a different taste from this process, and yeah, the blackthorn salt will have to come out at some point.
Yeah, no, I misspoke. I mixed up my Boston towns. It's not Malton. It's the one you said, John. It's
Yeah,
Medford, right?
Malden,
Yeah,
it's great stuff. Yeah,
yeah. No, I think I said Medford. I meant
yeah,
Malton.
yeah.
Yeah, no, it is great stuff. The flakes are phenomenal. But I might need to go offline and learn more about this Scottish salt. But yeah, no, we're going to wrap up, Brad. This is our last question for you. It's one that John and I always talk about. What are your? I know we got Christmas upon us here, but you got any weekend plans? That's usually what we talk about. Or did you take any good trips recently that you want to share?
Yeah, Dom, I'll just share the weekend plans. Actually, it's going to be a great Christmas. My daughter's coming back from college. She's at the military academy at West Point, so we
Oh
don't get to see
wow!
her that often. We may see her 2 weeks, maybe 3 weeks out of the summer, and she's actually on the lacrosse team as well. So she
Oh
she has a
wow!
full schedule. So we do visit her occasionally, but we have to go out to the games to visit her, and it's usually just an hour or so that we get
Oh
we
yeah.
get a snapshot. So mom's extremely excited about that. She gets 2 weeks with her oldest, and the girls will be together. So it'll be prepping for Christmas. Me wrapping the gifts, my gifts to the family, etc. You know how that goes, and
Oh yeah, I'm
so
all done. I love having them under the tree. Well, Brad, that's an incredible accomplishment by your daughter there. What year is she at West Point?
she's a junior this year.
Wow, and she's
So,
playing lacrosse. Well, I'm going to tell her thank her for her service because I think she's technically in the military as of her freshman year, right? And I have heard you get very limited time with your parents, so that it feels like you're begging for droplets of time with them, I suppose. But that's exciting, Brad, and I'm sure your wife is psyched, as are you. Well, that's awesome, John. What you got going on?
Well, it's well. You talked about trips we did. I mean, obviously around EBF, I had the trip to Edinburgh and stuff, which was fantastic. And Brad, when you talk about being a steak person, I'm more of a fish and seafood person. So going back to Scotland is a great opportunity to sort of get some really good fish cookery, and some of the restaurants in Edinburgh are excellent. This weekend coming up, it's getting prepped for the kids descending. So my wife and I both have our separate lists in terms of what we're baking ready. So we have food over the Christmas period. I had like one shopping list. My wife has got multiple pages of where she's going. So it's just that getting prepped. It's like I'll do an alcohol run of some things that we would only buy in at the Christmas period, and just looking forward to getting to Christmas Eve and having a break and just chilling towards New Year.
Yeah, and I think you even got to spend some time with some old high school buddies or
Oh
classmates.
yeah, that was fun. Yeah. I think did I talk about that last podcast?
I don't remember, but
that was basically I mean, one of my we got together and one of my friends worked out it was actually 50 years since we first met each other. So this was our like I say, our 50th year of getting together, and obviously I'm the one who my three friends have stayed around the Glasgow area, but they're still fairly scattered. I'm the one that's gone obviously thousands of miles away, so I only get back occasionally. They get together much more often than I would. But I've tried certainly with the recent years to try and get back together with them a bit more often. And this time, rather than meeting up in Glasgow and sort of doing various pubs, etc., we just met at one of my friend's houses, and he's worked for a number of distilleries and has a nice stash of whiskey in his attic, a lot of which is very collectible whiskey, but actually it's you don't just want to collect it, you really want to drink it. So he rolled out some very nice whiskies for us all to sample, and it was one of those things that got me in the Christmas spirit a month ahead of Christmas, because it was so nice to get together with friends who obviously, like say, we go back 50 years, we got a ton of time for each other, and it was just the nicest day we all spent together, and the whiskey helped, but I'd say it was actually the friendship was the key piece.
Yeah.
And it's almost like now I'm looking forward to doing the same thing November next year already, because it was such a nice experience, So
yeah. John, I suppose when you're not making eggnog with brandy or anything like that.
Our Christmas drink that we found years ago is actually a Nigella Lawson recipe. It's a drink called a poinsettia, and it's basically it's cranberry juice, Cointreau, then like a fizzy wine, like a Prosecco or a Champagne or whatever, and I think that's it. And so it's a bottle of fizz, a volume of Cointreau, and then a bunch of cranberry juice, and it is what, like you talk about the grapefruit beer, it is so drinkable.
Yeah, that sounds dangerous.
We've had it for Christmas for years, and my eldest son actually had it as his signature cocktail at his wedding back in 21. So we've got our supply. The only thing we don't have right now is the cranberry juice, but that'll be our Christmas Day feature.
Yeah, I don't do anything special for Christmas, but New Year's parties, I'm notorious for breaking out a big, kind of spigot-like container that holds about 10 gallons that I make a holiday punch in, and probably break that out this year. And it definitely livens the party up, because people don't realize how much alcohol's in there.
Sure. Yeah.
Because it's a whole process. I got to cut a whole bunch of fruit up, and
that's a dangerous thing. I remember once when I was in an apartment in North Carolina. So this goes back about a long time, and the the management decided to have a party in the apartment complex clubhouse, and for whatever reason, I don't know why, they decided that they were only serving spirits, and it was that same deal where people don't realize how much they were drinking. It was it degenerated into a complete shambles and fist fights and all sorts of stuff.
Oh, jeez.
It was one of those things, and I think I left early, and my wife missed me leaving, and she's like really worried as to, with all this stuff going on and knowing my tendency to get involved in stuff I shouldn't, she's like getting really worried where I am. I'd gone back to my apartment, gone to bed, and I was asleep and oblivious. But yeah, we had other friends who got a little bit more involved than I did. So ever since then, I've always been really cagey about doing too much spirits, and that's really why I don't drink shots, because if I'm drinking beer, I want to know what I'm drinking. I don't
idea, John.
mix something and go in the direction I don't want to be going.
Well, I think when in my world it's not quite as lively, but there's a karaoke machine and like some table games and stuff. And the first hour, as everybody's getting acclimated, it's really low volume, and then as the night wears on, the volume in the room gets louder and louder. And then by midnight, it's just it's a lot of fun, and everybody gets a lot of liquid courage to go up there and sing whatever song they might want. So maybe I'll wrap up. I got 2 older brothers and a younger sister. My 2 older brothers, I have a nephew that lives here, so one of them came to visit. They surprised me. They let me know about 5 days ago they're going to be in town. They're actually in town. Arrived yesterday. Got to spend a nice day hanging out with my brothers. And one lives in Colorado, the other in Massachusetts, and so they're here till Monday. So that's got my weekend kind of busy. We got some exciting stuff going on. Going to host everybody over here on Sunday, but we got like a Mavericks hockey game we're going to go to, which is should be fun. And those of you that do or don't know me, I'm the tall, skinny one in my family relative to my brothers, and I'm actually not even the loudest of the 3 of our siblings. One of them is quite the gregarious individual, and he's just like a walking riot, and he's the Bostonian, so you often see a lot of surprised looks from everybody he talks to, because he's so engaging. And I think Midwesterners are kind of like, is this guy going to mug me, or is he just a happy individual? So it's quite fun to watch and to be in the family, and really I relish it and cherish it. And really, right after this podcast, I'll go see him. So with that, John, I don't have anything else. Brad, any last parting thoughts here as we wrap up?
Yeah, gentlemen, no, a big thanks again for allowing me to come on the podcast. It's great. We had great science, great food, great drink. We're going to have a great time with our families. And I would really love to see some video of Dom, your family up on that karaoke machine after a little bit of that liquid courage. It'd be fun to share. Maybe you can grab some highlights on your next podcast. I think the listeners would surely enjoy it.
If you don't know, KCAS has a few like professional singers that are working for us. So they'll show up and they put on great shows, and my wife loves to sing. But my go-to song is the Humpty Dance, and it's quite entertaining. I've got that one down. I know it's not probably the most, it's not the best song, but it's one that gets the crowd going, and it's quite funny to see me up there trying to rap. Usually I'm sweating by the time it's over, but it's a lot of fun. And, John, I don't have anything else other than my trademark is I'll see you in the funny pages.
Yep. Nothing else to add. Thank you very much, Brad. And I really enjoyed this. Thanks a lot.
Gentlemen, thank you.